In case of the gene expression, it is a broad term. Why it is considered as a broad term? Because it has to encompass different molecular process. And finally, it is going to give the end products of the gene, that is the RNA and protein. Suppose if you consider the gene expression as a protein production, that is a polypeptide formation, it exerted at various levels, that is the 4 levels in case of eukaryotes. Because in case of the prokaryote, the transcriptional initiation itself a predominant target site for control of the gene expression. Because the translation occurs in a single compartment. There is no demarcation of nucleoplasm and cytoplasm. Why? There is no nuclear membrane. So in case of the eukaryote, it exerted at 4 levels. The first you see, this is the transcription level. Transcription level. So in case of the transcription level, different regulatory proteins affect RNA polymerase in detecting the start site. What are the start site? The structural genes. And the second, processing level. So the first RNA formed, the initial transcript is going to have exons and introns. The introns has to be deleted, exons has to be joined in a particular fashion. Because the alternate splicing is species specific, time specific, tissue specific. Suppose if we take the example cytochrome C, cytochrome B, C1. Cytochrome B, C1, it is the component in case of the complex 3. In case of cytochrome C, it is a carrier between the complex 3 and complex 4 in case of the mitochondria. So, cytochrome B, C1, the protein required in case of the complex 3. Whereas cytochrome C, it is required in between the complex 3 and 4. So, it is the time specific. So, alternate splicing, it is required. So, different exonic sequences are present in case of the processing level. Transport level. After processing, the mRNA has to be protected. So, in order to protect this mRNA, at 5 end, there is a capping. 7-methylguanosine triphosphate is added to this 5 end of hnRNA in a template independent fashion. It protects the cleavage of RNA from 5 end to 3 end. Next, in tailing, 200 to 300 adenylate residues, that is adenine nucleotide, added at 3 end in template independent fashion. So there is a processing is required in order to transport from the nucleus to cytoplasm. Next, this is translation level. After formation of the protein, so as to become a suitable protein, it has to undergo so many chemical modifications. And the organelle concerned with the chemical modification, the glycolization, glycosidation, that is the Golgi complex. So final destiny of the protein is decided at the Golgi complex where the protein has to be transported, whether the plasma membrane or the lysosome like this. So gene expression exerted at the 4 levels in case of the eukaryote.